The first step, as we talked about, one ATP is getting consumed through the action of hexokinase. We are getting glucose 6-phosphate. Hexokinase gets its name. Hexo means 6. There are 6 carbon atoms. And kinase, they are taking phosphate from ATP and they are putting it here on carbon number 6. And that's why we have glucose 6-phosphate. And our glucose 6-phosphate undergoes isomerization, and what we have is going to be fructose 6-phosphate. Fructose 6-phosphate, through the action of a very important enzyme that's called phosphofructokinase, gets converted to fructose 1,6-bisphosphate. Why is it called that? Because we have two phosphate groups, one on carbon number 1 and one on carbon number 6, and that's why it's called 1,6-bisphosphate. Why do we call it bisphosphate? Because the phosphate groups are far apart from each other. This step is the second step that we are using ATP. Our ATP is getting hydrolyzed to ADP and the phosphate group is coming here. The next step is going to be an aldol cleavage, which is catalyzed by aldolase. This molecule is getting cut into half. We are going to have dihydroxyacetone phosphate and we are going to have glyceraldehyde 3-phosphate. And what happens next is that our DHAP gets converted into glyceraldehyde 3-phosphate through action of an enzyme that's called triose phosphate isomerase. So by here, we are going to have 2 mole of glyceraldehyde 3-phosphate that has been made from 1 mole of glucose. From here onwards, each reaction is going to happen twice because now we have 2 of this molecule. The next step is going to involve an enzyme that is a dehydrogenase, glyceraldehyde 3-phosphate dehydrogenase. Dehydrogenase is an enzyme that is catalyzing a redox reaction, which means our glyceraldehyde 3-phosphate is getting oxidized. In other words, it's losing electrons, and these electrons are getting transferred to NAD+, which is getting reduced, or it's gaining electron and it's getting converted into NADH. There is an addition of inorganic phosphate that gets added here as well, and the molecule that we end up with is 1,3-bisphosphoglycerate. Now, in the next reaction, we have an example of something that we call substrate level phosphorylation. Substrate level phosphorylation. We are getting production of ATP without going into the electron transport chain. So what's happening here is that our 1,3-bisphosphoglycerate that had 2 phosphate groups on carbon number 1 and on carbon number 3 is getting converted to 3-phosphoglycerate, and this phosphate on the carbon number 1 is going away. It's getting transferred to ADP to make ATP, and this is going to happen through the action of phosphoglycerate kinase. And as we said, kinases are the enzymes that are going to move phosphate group into ATP. What happens next is through the action of phosphoglycerate mutase, we are going to be removing this phosphate group and putting it on carbon number 2, and the new molecule is going to be called 2-phosphoglycerate. And after this, through the action of enolase, a high energy enol bond is going to be made and 1 molecule of water is going to get released. The molecule that we have is going to be called phosphoenol pyruvate. Phosphoenol pyruvate has a phosphate group on carbon number 2 that gets removed through the action of pyruvate kinase. This phosphate group is going to go on ADP to make 2 molecules of ATP.